scientist and the lab manager for 4Life. I have been working for 4Life for almost 4 years now, and let me tell you, it has been a wonderful place to work. Let's see if I've got my slides. Yep. I love being part of a company that cares so much about the importance of providing safe, effective, and innovative products. And 4Life is committed to creating the best products on the market with proven ingredients and definitely have the science to back them up. So today I'm excited to share with you all some of the fascinating science behind our transfer factor. After all, the more we understand transfer factor, the more we can use it to make life better for people around the world. So here in R&D, we get a backstage look at the research that's been done on transfer factor in the immune system. We're always on the lookout for new and exciting experiments that we can do to both validate what we already know and to provide new insights in how transfer factor works. Every experiment that we do gets us one step closer to understanding everything we want to know about transfer factors. So let's dive right into some science. Today I'd like to talk about an experiment that we've been working on recently. Whereas some of the transfer factor research we do is in-house with little help from prestigious universities, the study I'm about to tell you about was designed in collaboration with the minds at 4Life and the scientists at Reach Bio Research Labs. Reach Bio focuses their expertise on cell biology and experiments based on cells, and that makes them a perfect choice for work with cells in the human immune system. So this experiment has been ongoing. We've done the work in phases, and every time we do the experiment, we learn something new and figure out ways to optimize our test results. We have developed this bioassay to validate the actions of transfer factor and other peptides on the functions of the innate immune system. The second important goal of this experiment was to create a quality control tool to verify the transfer factor's potency. We would all like to be assured by objective testing that our products actually do what we say they do. In addition, we wanted to look at our different sources of transfer factor: ovofactor, nanofactor, and of course, ultrafactor. Finally, we wanted to compare the activity of our transfer factor products with those of our competitors. We want to see who has the best supplements. So this assay was conducted on a very expensive and complex instrument called a flow cytometer. It can sort and count individual cells based on their characteristics. In fact, it counts every single individual cell one by one. This provides excellent data that we can use computer software to analyze. This is a method that many industries use to explore all types of cellular functions. It can be used to evaluate drugs, botanicals, natural products like our colostrum, and many more at a cellular level, giving us a very close look at what happens and how it works. Since the scientists at Reach Bio are the experts, we relied on them to help us run the instrument as well as design the assay and interpret our test results. So let's talk for a minute about how the test actually works. In brief, the experiment uses natural killer cells, or NK cells, from healthy blood donors. It incubates those cells with the compounds that we want to test, allowing those compounds to activate the cells. After those activated cells are exposed to the compounds, we then expose them to target cells that represent a threat to the human body. This image shows what it looks like when an NK cell attacks a target cell. Now, after the NK cells are activated by the compounds, we give them a chance to kill the target cells, and then the target cells that are left over that don't get killed are counted and sorted on the flow cytometer, and then we get our test results. So we tested several different kinds of samples. First, we tested multiple batches of ultrafactor from different seasons and different dairies to see how much variability there is from lot to lot. Then we tested the individual components of TriFactor to see how much each component contributes to the overall effect. Next, we tested colostrum-based products from our competitors. So let's look at some actual test results. This is an example of a flow cytometry readout. I know it looks really complicated, but all you need to know is that one dot represents one single cell. So on the left-hand side, you can see the NK cells in red and the target cells in blue. And on the right-hand side, after the transfer factor was added, you can see that the red NK cells killed almost all of the blue target cells after they were activated by transfer factor. In fact, almost 99% of the target cells were killed. Isn't that amazing? Now make sure you guys come to the workshops on Friday, on the day of learning. Dr. Paula Brock, who we just heard from, is going to talk about this experiment a little bit more. So let's make some sense of those flow cytometry readouts we just looked at. We can use computer software to translate the pictures into actual numbers, and numbers are easier to see and explain. So let's take a look at some data. In this experiment, we used interleukin-2, or IL-2, as a control, which means something to compare to. IL-2 is a signaling molecule that we already know activates cells, so it's kind of the gold standard in this type of experiment. We compared IL-2 to ultrafactor, and here's what we found. The natural killer cells that were exposed to ultrafactor killed 3 times as many cells as those exposed to interleukin-2. That means they were activated 3 times more effectively. Okay, what other results did we see? Here, we took a look at the lot-to-lot variability of the ultrafactor XF ingredient. We tested 11 different batches. They were from all different seasons of the year and all different locations. And as you can see, there was a bit of variability, but overall, we got pretty consistent results. Basically, what this tells us is that every batch of transfer factor that we put in our products is going to give you the same great benefits. We also tested TriFactor and its 3 components, ultrafactor, ovofactor, and nanofactor. What we found was the combination of the 3 outperformed each individual ingredient, and that shows a synergistic effect from combining the ingredients together. We also looked at a few of our competitors' colostrum-based products. As you can see, all of them did have some activity, but all of them underperformed compared to 4Life's products. So as you can see, we really do have the best immune system supplements. So to summarize, we got a first-hand look at the strong effects of ultrafactor, the lot-to-lot consistency of the ultrafactor ingredient, the effect the different components of TriFactor have, and the synergy created by adding them together, and how we compare to our competitors. So all in all, I would say this was a pretty successful experiment. Now, before I wrap up, I want to share just a little bit more. 4Life is always looking for new and exciting ingredients to put in our products, and in the future, we can use this same experiment to investigate their effects on the innate immune system. So we're always striving to further our knowledge about our ingredients, especially the transfer factor, so we have many other experiments and studies in the works at all times. There are countless ways to investigate transfer factor's effect on the immune system, and this can be accomplished by experiments that measure all sorts of biomarkers, including genes, enzymes, signaling molecules, and many more. One goal that we have here in research and development is to isolate the transfer factor on a molecular level. We want to see what it's made of. We do already know that it's a peptide, which is just another name for a small protein, but we'd like to know how the molecules are actually made up. One way to try this is called chromatography. It sounds complicated, but all it is a way to separate the transfer factor from the rest of the colostrum. Once the molecules have been collected, we can do experiments on them to learn more about them. These are just a few ways that we can keep learning about transfer factor. We're always looking for new ways to learn, so stay tuned for future research. Thank you so much for listening. Thank you for coming to For Life's Convention 2019 RISE.
Servicios de traducción en español, canal 74.7.